going to introduce one of the last keynote speakers, Dr. Evgeniya Kirilina. So Dr. Kirilina received a PhD in MR physics from the Russian Academy of Sciences in Novosibirsk, Russia. After PosDocs at the Free University in Berlin, she became a leading MR physicist at the Center for Cognitive Neuroscience in Berlin. She now heads the MRI Biophysics Research Group at the Department of Neurophysics at the Max Planck Institute for Human Cognitive and Brain Sciences. Her goal is to develop novel non-invasive methods to map brain iron with high sensitivity and specificity for cellular distribution and chemical form to understand the contribution of iron to brain development and neurodegeneration. Today, she's going to talk more specifically about mechanisms of iron-induced brain MRI contrast in Alzheimer's and Parkinson's disease. So please, Dr. Kirilina, you can now share your screen, and I'm very excited to hear you talk.
Thank you very much for this kind introduction and your kind invitation. Can you see my screen now? Yeah, fantastic. Thank you very much. I'm enjoying this conference very much, and it's a great pleasure to be here. And it was exciting talks so far. So I wanted to quickly show you my group members who actually did the most of the work I'll be presenting, and then I'll start. So if we think about the brain structure, and especially in the context of neurodegeneration, then we can describe it at the different spatial scales. So on the macro scales of centimeters, we can look at the changes on the brain macrostructure, and this is where the standard MRI works very well. With the development of the ultra-high field, we are now able to resolve very small structures such as cortical layers or tiny brain nuclei, and that's already very exciting. But what would be extremely important and helpful is to be able to detect microstructural differences and to detect cells or fibers. And this is only possible when we apply biophysical models of the MRI signals. And I think we have seen fantastic examples yesterday in Marcos and Ricardo's talk in the context of diffusion. And I will show you how this can be done also for relaxometry and particularly for mapping of brain iron. And why is it possible? It's because water, which we actually observe most of the time in MRI, probes and interact with the tissue or brain tissue at the very different spatial and temporal scales. And actually, most of this circles here has been fantastically covered by previous speaker, with Aviv talking about the molecular interaction at T1, Carl talking about the MT, Alexander looking at the water content and the water in the myelin, and fantastic insights from diffusion yesterday from Marco and Ricardo. So the only things which actually left is their magnetic inclusion, and this is the topic today. So one of the most prominent magnetic component of the brain tissue is actually iron. And iron is a transition metal with the unique catalytical properties. It has 5 unpaired d electrons in the outer shell, and the electronic structure of and actually its magnetic properties strongly depend on the local environment of iron. And it makes it very reactive, catalytical, and magnetic ion. So first of all, usable in catalysis, but also visible in MRI. And this is why most of the cells in our body use iron, and particularly brain cells use it for as a cofactor for the neurotransmitter synthesis as a dopaminergic neurons. In myelination, for oligodendrocytes, it is used to the myelin synthesis. And finally, metabolically active microglia need a lot of energy, and this is why they also rely on a high iron level. However, when present in a high concentration, this highly catalytical property become harmful and can induce oxidative stress and even lead to the cell death of the mechanism of ferroptosis, which was recently discovered. This is why iron is very carefully regulated in the brain. Its intake is regulated by the transferrin receptor and the transferrin. Then the storage, so it's stored in the ferritin or in neuromelanin in dopaminergic neurons. And the release from the brain is much slower than the iron uptake to ensure this high level of iron. This is why iron typically accumulates with age in the brain. And this is when it becomes harmful, because iron overload in age was associated with a faster cognitive decline and is particularly observed in the context of Parkinson's disease and Alzheimer diseases. Therefore, monitoring of brain iron non-invasively is very important and has a high diagnostic potentials. So it has been realized very early, already in the 80s, that iron is sensitive so MRI is sensitive to tissue iron, and by comparing spatially their T2 weighted images in vivo with the postmortem stains for iron, people realize that a lot of contrast, especially in the basal ganglia, is actually mediated by iron. And this contrast is even stronger when we go to the ultra-high field, and he's one of my favorite papers on iron is from Jeff's Dunn group, showing that the intracortical layered contrast is actually induced by the combination of the iron and myelin contrast by comparing it with histology. However, here the validation with histology, particularly the context of iron, has its limitation because histology is not quantitative, and MRI has a potential to quantify iron. Therefore, later, the people started to combine advanced methods of iron quantification in the brain with MRI, and here's a beautiful work by Carsten Stüber, who combined proton-induced X-ray emission for quantification of iron and myelin and demonstrated that there is a general linear model can be created to describe the R2 star as a combination of two. The very important work by Christian Langhammer implied the same methods in their context of quantity susceptibility mapping. And more recently, there is a very impressive work of Simon Hammondner who combined actually histology in 3D over the whole brain, which he calibrated by the laser ablation in different small, with the small pieces, actually point by point. However, all of this method actually have a mostly empirical linear model of total tissue iron and doesn't account for their molecular form and cellular distribution, more importantly. And if you think about the different forms, and I just shown here the few, and I think also Aviv was showing this introduced this different form in his talk, that there is a iron magnetic moment is very, very different per iron atom, depending on the form of iron binding. For example, it's almost double for transferrin as compared to ferritin. And for some of the form, we actually even don't know because iron is distributed between the two different binding forms. Moreover, from physical principle, we can show that also cellular distribution should matter, because if we think about the mechanism of iron-induced relaxation, then it also happens in different spatial and different temporal scale. The first, in the nanoscale, we have a molecular interaction of the ferritin molecules with water, and this happens very, very quickly because the diffusion average most of this contribution. So we are in the so-called regime of motional narrowing and have only T2 contribution from this mechanism. On the other limit sides, on the meso and macro scale, of course, cortical layers, vessels, or overall bigger structures of the brain also induce variation of the Lamont frequency and cause R2 prime static dephasing. But the most interesting part is, and it's actually the major contribution to in the brain, is happened on the micro scale of the structures, iron-rich structures in the size of several micrometer. They are not fully averaged by diffusion, and they contribute strongly to R2 star. And this is actually depending on the size and the iron load of the cell. We would have a different balance of R2 and R2 star contribution, and it makes it sensitive to the cellular form. And here, just to give you an example, we can see that the iron load and the shape and the density of the cells is very different between these known iron-rich cells. And this is exactly what I would like to explore together with my group to develop iron markers with the cellular specificity. And that's the overall avenue we are following. So we combine ultra-high resolution MRI with the in vivo, with the postmortem microscopy, where we can apply advanced method of iron quantification with cellular sensitivity and cellular resolution to create biophysical model of magnetic field perturbation within the voxel, which we then can validate and then finally apply back to in vivo to create the co-MRI markers of cellular iron. And I will show you two examples now of two different forms or two different roles of iron actually. And the first is iron in neurotransmission, how we can use quantitative MRI to map dopaminergic neuron density and iron load. And this is actually a work by a PhD project of Marta Bramolo, who will graduate just a week after next week. So dopaminergic neuron cells are the pigmented cells located in their substantia nigra in the basal ganglia. And the loss of these neurons is the primary cause of motor symptoms in Parkinson's disease. The cells are very special, so they are very large and they are pigmented and they are organized in so-called clouds, so-called nigrosomes, with the nigrosome 1 being the largest here and the affected earliest in the PD. And unfortunately, at the moment of the first PD symptoms, motor symptoms, 75% of the cells is already lost, so there are no curative treatment for PDs possible. Therefore, detecting the cell loss at the early stage would be a fantastic and very important and a prerequisite for PD therapy. And iron play a very special role in the cells because these neurons are very, very iron-rich. And the reason for it is that they require iron for the synthesis of dopamine. The iron is a cofactor in the tyrosine hydroxylase, and therefore the high level is requested. But the combination of dopamine and iron is a very neurotoxic thing, and actually the oxidation of dopamine by iron can lead to the generation of ketones, which are very toxic. Therefore, there is a special neuroprotective mechanism of creating such a polymer, neuromelanin, which combine lipids, iron, and ketone polymerization products. And then they are microautophagy, they're so-called neuromelanin organelles formed by the cells. And this accumulates in the cell with age, and probably overload with neuromelanin may contribute to the vulnerability in the Parkinson's disease. And here, the special thing about the neuromelanin that the iron bounding is very complex. So there is at least two different sites. One is the neuroprotective, the crystalline form, which has a high affinity and safely stored safely there. But there is also neurotoxic binding form with a low affinity, which is paramagnetic. So they have a very different magnetic susceptibility also. So our first step to understand the MRI contrast by neuromelanin and try to detect the biomarkers of the cells was to understand how actually the one single cells impact the MRI contrast. And for this purpose, we use the postmortem MRI microscope at ultra-high field combined with ion beam microscopy. So you see here gradient field echo image acquired at 22 millimeter resolution at 9.4 Tesla from postmortem tissue. And these dark dots in their magnitude and this dipole-like shapes in the phase image actually correspond to single neurons. And we can allocate these neurons and determine the iron concentration in them after we perform the MRI scanning. So we can basically use it with to simulate the signal using the magnetic sphere model, accounting for diffusion and partial volume effect. And here you see the simulation, and that's an experiment. So we can determine for each individual cell, we can determine its magnetic moment from this data. What we can do next is to plot the cellular magnetic moment of each cell as a function of its cellular load, and then with a linear fit determine the susceptibility of neuromelanin, as we have done here. And interestingly, it's also 3 times nearly larger than the one in the ferritin iron. Already this value is very interesting because it's actually showing us that it's much closer to the neurotoxic binding site susceptibility compared to the neuroprotective. So it may be that actually this is a dominating form, at least in an old age, and that may explain the high contribution of iron to PD pathology. But now, knowing the susceptibility, we can go from the one cell to individual voxel and create the model of our R2 star informed by cellular iron distribution. And what we have done, or what Matt has done, he created the 3D iron concentration by map using the subsequent physiological sections calibrated with a proton-induced X-ray emission, and then simulated local magnetic field perturbation around each cell. So that's basically the model of the voxel here. And then accounting for diffusion in the Monte Carlo simulations, he was comparing the Monte Carlo simulations, properly accounting for diffusion, with the static dephasing prediction. And for this particular cells, with their very high iron concentration, actually static dephasing holds very well. And this allowed us analytically predict their contribution to R2 star. And now what is nice about the simulation that we can now calculate the contribution of different forms of iron and the different relaxation channels to iron-induced relaxation. And this is shown here on this busy slide. So you see that's an overall relaxation. That's the one, the red one is the one induced by iron, and this is other sources. And here there are different contributions from the simulation. And what we have shown that actually 12% of iron and dopaminergic neurons, so they only contribute a very small percent of overall iron, induced 60% of R2 prime in nigrosome 1. So this means that it may be a potential quantitative biomarker of dopaminergic cell density and iron load. So we were very excited and tried to look how it can be applied in vivo, but when we look into the radiological literature, then we were surprised because actually the current interpretation of MRI contrast in substantia nigra completely contradicted our biophysical model. Because actually it has been shown that their so-called swallowtail sign, that's a hyperintense part of substantia nigra, disappear in the Parkinson disease, and therefore it was associated with the nigrosome 1. And our predictions say nigrosome 1 should have a very short relaxation time, so it should be dark. So it's basically completely inversion of the contrast. The difficulty here is that the structures we are talking about is very tiny, and this interpretation was based on one single study with a 2D histology, and the structures are really tiny. So it's like a bean, and they're the ruba is just like a pea. So you need a very precise histology and a perfect co-alignment of signals to co-align the structures correctly. And here we used the unique opportunity of the collaboration which we have with a group of Birte Forsman and Annika Alkemade in University of Amsterdam, where we collected the combined data set combining the several quantitative MRI parameters and multiple histological stains in their several postmortem brain. And that's a 3D histology on the many brains. And what we have done then, we did an independent segmentation of the swallowtail sign and their MRI, and the used postmortem histology to look at the nigrosome. And what we have shown actually that these two structures co-align, but only partly. Actually, the nigrosome has a very different shape and is much longer than ultrastar. So also they come together, they're actually not identical, and the presence of one doesn't mean that that's and the byproduct of this work was actually the creation of the Nigral Atlas, and that was our COVID pandemic task, where we manually segmented a lot of images and now want to release this data set and as a part of the lead DBS open resources. So, but I think that's stress that the 3D histology, as has been also shown yesterday very nicely, it's very important very often for validation of MRI. And now I would like to show you another example to switch the gears here and talk about the iron in the chronic inflammation, so iron-induced contrast in the context of Alzheimer's diseases. And we particularly looked in one specific form of Alzheimer's disease, posterior cortical atrophy. This is atypical Alzheimer's disease, which starts in the visual cortex, and it affects their occipital lobe first, but also as Alzheimer associated with the amyloid beta plaques. And you may know that abnormal brain iron accumulation is reported in Alzheimer's disease, both in the macroscopic level, but also in the microscopic level. So there is this known high accumulation, but also the strange distributions were reported. And it's not completely actually known if the iron is here the pathogen or symptoms or both. So that's one of their schemes I adopted from the recent review, where that's a current view that due to the genetic risk and aging, accumulation of amyloid plaques happens and the blood-brain barrier breakdown. And due to this fact, iron-rich macrophages and microglia invade the brain. And then this leads to the increased level of labile iron, oxidative stress, and neurodegeneration. But it's not quite clear which stages, so this overall iron we measure with MRI, which stages of this it reflects. And the goal of our study was here to establish the relationship between the macro and microscopic iron measure and actually see if this microscopic relates to the amyloid plaques. Again, we took the post-mortem tissue from several patients with the posterior cortical atrophy and with the typical Alzheimer's disease and compared them with control. And what we have seen, there is this characteristic hyperintensity in T2 star weighted images or hyperintensity in R2 star can be observed in the brain of patients. However, overall values in the cortex didn't differ significantly from the control brains. And then we performed the iron quantification using X-ray fluorescence here and found the surprising results. So we found that the hotspots of iron accumulation in Alzheimer cortex actually didn't correspond to the position of the amyloid beta plaques, as had been suggested by some previous literature in the animals. We confirmed this by the double stains, and here you see the iron stained in brown and the plaques stained in blue. And also, if you look at the stains separately, they show very similar distribution. Actually, they don't co-align on the local. If you zoom in into the individual plaques, here again stained with the nickel, and here just a natural iron concentration, then we have actually seen some hotspots of iron accumulation in the plaques, which resemble the shape of microglia on the sum of the plaques, also visible in the stains. But now we can do again biophysical modeling. So here you see actually the analysis of the big population of plaques here. So they were segmented, and then we quantified the amount of iron in the plaques, and then in the closest surrounding around them. And you see it in this plot. So this is a median of the plaque concentration. This is surrounding tissue. And also there is a slight elevation of iron here. It's really tiny. So it's only 15% more than in the surrounding tissue. And that's due to the presence of this microglia in some of them. So if we now estimate how much this iron in the plaques contribute to the R2 star, and it is shown here, the prediction of the theory in blue and red plots, it's really tiny. So this iron enhancement is not indicative for plaques, as had been suggested by some animal literature. So it's probably reflects something else, something like leakage of the vessels or something else. So here's a small conclusion that the hotspots of iron accumulation in Alzheimer disease and PCA are not in amyloid plaques. Iron in the plaques constitutes a negligible contribution to T2 star contrast, and the elevated iron levels in PCA and AD are not indicative for plaques, but may reflect inflammation of brain blood barrier leakage. And here I'd like to summarize my entire talk, saying that the iron-induced relaxation rates R2, R2 star, and QSM are sensitive to cellular distribution and chemical form of iron, and the biophysical models of MRI contrast induced by dopaminergic neurons and amyloid plaques has been created. I think they're useful to understand sensitivity and specificity of our markers, and I hope I have shown you that the specific information of brain iron can be obtained with MRI. And with that, I would like to acknowledge my group, Neurophysics Department, and funding and collaborators. And thank you very much for listening.
Thank you so much. It was an amazing talk. I really appreciate it. Mark, do we have any questions in the chat?
Yeah, we do. We have quite a few. I'll jump right in. From Dr. Hagberg, I think this question is directed at the first part of your talk on Parkinson's. She asked, what about the validity of these beautiful ex vivo observations for clinical in vivo MRI?
Yeah, that's a very good question. And I didn't put it in my talk, but we do a lot of work to try to address some of the limitation of the postmortem work. And there are several. One is, of course, just if there are probably, so if their tissue cellular distribution changed by fixation. I think it has been addressed by some of the previous speakers. So what we do is that to follow the fixated brain just very briefly before death and then over the time of fixation. And we have really observed one very spectacular case, and that was the case of locus coeruleus, where we observe a 10-time increase of iron concentration inside of the cell. So it can happen that, for example, if there's some gelatin compound in the cells, that you can have a really redistribution of cellular iron. We haven't observed it in the SN. Another big limitation Actually, let me see if I can show you a couple of slides. So is that we always have the post-mortem research is performed on older humans. So extension to the lifespan is very, very difficult. And the common animal models are not helpful because, for example, in case of neuromelanin, they just don't contain neuromelanin. And you need long-living animals to do so. So we do the validation. They're not in rodents, but we have a unique possibility to look at the tissue of chimpanzees because there is a project where we collect the naturally deceased chimpanzees in the wild or in the European Zoo, and then they're brought to Leipzig. And there we can actually extend the models to the entire lifespan. And here you see, for example, accumulation of neuromelanin in the chimpanzee brain here. And that's something which we really even can do in humans. And to make a long story short, for example, here's the applicability of the model we performed in the chimps of different ages. And we have shown that at the age of puberty, probably this biophysical model should work. But you're completely right. It's very, very much keep in mind that postmortem is postmortem. And therefore, all of these fantastic approaches have been shown yesterday also with the in vivo models, and age effects are also very useful.
Okay, thank you. Christopher Rowley, he asks, he says, great talk with some very beautiful images. You found that the iron magnetic moment aligned closer to the non-protective iron form in the neurons. Could this be because the damaging iron is the iron that isn't being shielded by neuromelanin and is free to react with surrounds such as water where it may be affecting MR signal more?
It really depends on the contrast. So one should say that the R2 stuff, it's an excellent question. So yes, first answer is due to the fact that it has a double magnetic moment, it will affect all of your susceptibility contrasts, R2 star, R2 contrast stronger. So it's exactly proportional to the susceptibility. I think there may be even more strong effect for T1 because as you said, the monoatomic iron is exposed to the water, diffusing water much stronger. Actually, I think that some of the neuromelanins and different MRI may be due to the fact that there is this really strong T1 shortener. We haven't looked at that theoretically. I think that would be really interesting. T1 is really tricky, like to create the generative model. But I think that would be really, really interesting to look at that as well.
Okay. Okay, thank you. Maybe we have time for one more. I think we started a few minutes into your time. From Olivier Perron, he says, would it be possible to disentangle the iron in oligodendrocytes from the iron in microglia to better estimate neuroinflammation?
That's a fantastic question. And I think that that's really something what we want to try. So oligodendrocytes and microglia, they are both kind of the intermediate size and have a different iron, they have a higher iron load. So they are in this difficult intermediate regime. So theoretically, they're a little bit difficult to describe, but I think with the good priors, one can try to look at it. So we are trying to look at it, and actually this monkey data are extremely interesting. So you see these red dots, it's actually oligodendrocytes. So, and you see this iron accumulation with age, which is, and also the hot spots are actually strong iron accumulation in the age where they are building a lot of myelin. I think it's really interesting. We should try it. It's probably difficult to be really specific there, but I think with the good spatial priors, knowing where to look at, I think it may be possible. I hope it will be possible.
Okay, thank you very much. That was very exciting. So we've got a few more here, but maybe we'll save these for the post discussion.
Yes, amazing. That was really great discussion. Thank you.